Hello everyone, my name is Alper Tunga Özdemir. I work as a medical biochemistry specialist at Merkez Efendi State Hospital in Turkey. First of all, I thank the organizing committee of Beyond Science Initiative for giving me this opportunity. In this presentation, I will talk about our study on mesenchymal stem cells and toll-like receptor interactions and the effect of this interaction on NK92 cell-mediated anti-tumor immunity. First, I would like to give some information about the toll-like receptors. According to Wikipedia, toll-like receptors or TLRs are a class of proteins that play a key role in the innate immune system. They are single-pass membrane spanning receptors usually expressed on sentinel cells such as macrophage and dendritic cells that recognize structurally conserved molecules derived from microbes. There are many isofunctional toll-like receptors, and they can be in heterodimeric or homodimeric form. Toll-like receptor 3, 7, 8, and 9 are localized in cytoplasm, and others are on the cell membrane. They recognize specific structures of invading organisms, such as lipopolysaccharides, flagellin, 3-acylated or deacylated lipoproteins, or genetic material like as double-stranded RNA, single-stranded RNA, or CpG DNA. Each immune cell type has own specific toll-like receptor expression profile. When toll-like receptors are activated, the secretion of inflammatory cytokines increases, like as interferon gamma, TNF-alpha, interleukin-1 beta, interleukin-6, interleukin-8, and MCP-1. In addition, an increase occurs in the cellular activities such as phagocytosis, chemotaxis, cytotoxicity, and ROS production. Interestingly, some toll-like receptors are expressed not only in immune cells, but also in tumor cells or cell lines. In this slide, I would like to draw your attention to toll-like receptor 3, 7, 8, and 9, because endogenous ligands like dsRNA, ssRNA, and CpG DNAs activate these toll-like receptors. Given the proliferation rates of tumor cells, cell death and necrotic core formation are inevitable. And these necrotic cellular debris are potential sources for dsRNA, ssRNA, and CpGs. Sorry. From this perspective, these endogenous ligands should activate immune cells and contribute to immune elimination of tumor cells. Can we increase anti-tumor immunity by using exogenous toll-like receptor agonists? In ClinicalTrials.gov website, you can find 69 studies when you search using the keywords toll-like receptor and cancer. However, 8 of them were terminated and 5 of them were withdrawn. We see that only 27 studies have been completed, and only few of these completed studies have results. For example, in a study about imiquimod, it's a toll-like receptor 7 agonist, and skin metastasis of breast cancer have been reported that only 2 patients and only partial response were obtained. In another study, a combination of motolimod it's a toll-like receptor 8 agonist and doxorubicin was used in ovarian cancer, and it was reported that this combination had no effect on clinical results. In another study, a combination of EMD, it's a toll-like receptor 9 agonist, and cetuximab was used in metastatic squamous cell carcinoma, and it was also reported that this combination had no effect on clinical outcome. Why didn't toll-like receptor agonists work? Let's take a closer look at tumor tissue to better understand this. Tumor tissue consists of different cell types such as malignant cells, necrotic cells, immune cells, and mesenchymal stem cells. The most prominent features of mesenchymal stem cells are their immunomodulatory effects of immune cells such as T cells, B cells, NK cells, and dendritic cells. Mesenchymal stem cells can suppress almost all immune cells in non-selective and non-specific manner by secretion of suppressive molecules like interleukin-10, prostaglandin E2, IDO, hepatocyte growth factor, transforming growth factor beta, etc. Interestingly, it has been reported that mesenchymal stem cells can express some toll-like receptors, and the immunomodulatory effects of mesenchymal stem cells may alter depending on the type of toll-like receptor stimulation. It has been shown that mesenchymal stem cells stimulated with toll-like receptor agonists have pro-inflammatory effects, but stimulated with toll-like receptor 3 agonists have an immune suppressive effect. If the type of toll-like receptor stimulation is an important determinant of the response generated by mesenchymal stem cells, could mesenchymal stem cells in tumor tissue be behind the failure in clinical trials? To find a response for this question, we performed a in vitro cell culture study. We cultured the cells in the transvial co-culture system. We used inserts containing 0.4 micrometer pores that allow prior interaction. To observe antitumor effects of NK92 cells on breast tumor cell lines, these cells are co-cultured directly at the bottom of the well. To observe effect of toll-like receptor 3, 7, 8, and 9 agonists on the NK92 cells, cells were cultured with specific toll-like receptor agonist cocktails. Finally, to observe paracrine effects of mesenchymal stem cells on the NK92 cells, mesenchymal stem cells were indirectly cultured in the upper chamber. After the incubation period, we carefully collect NK92 cells and perform qPCR analysis to evaluate changes in specific inflammatory and cytotoxicity gene expressions. In this slide, there are graphs of NK92 cells co-cultured with MDA and B231 tumor cells. As can be seen, interferon gamma expressions increased up to 12-fold, TNF-alpha up to 10-fold, granzyme B up to 12-fold, and perforin up to 7-fold by toll-like receptor 3, 7, 8, and toll-like receptor 9. However, these increase significantly reduced by presence of mesenchymal stem cells in the blue line. You can see in the blue line. And reduction in cells stimulated with toll-like receptor 3 and 9 agonist was more pronounced. In this slide, there are graphs of NK92 cells co-cultured with MCF7 tumor cells. Similarly, interferon gamma expression increased up to 12-fold and TNF-alpha up to 6, granzyme B up to 5, and perforin up to 3-fold by toll-like receptor 3, 7, 8, and 9 agonists. Interestingly and similarly, these increases significantly reduced by presence of mesenchymal stem cells, and reduction in cells stimulated with toll-like receptor 3 and 9 agonist was more pronounced. In addition to gene expressions, we evaluate specific NK cell activation markers by flow cytometry. As can be seen, histogram changes of NK92 cells stimulated with toll-like receptor 7 and 8 showed minimal fluctuations compared to cells stimulated with toll-like receptor 3 and 9. In the comparison of median fluorescence intensity values, we found significant decreases in CD69 and CD107A levels of NK92 cells stimulated with toll-like receptor 7 and 8. In MTT analysis, we found that the cytotoxic effect of NK92 cells on MDN and MCF7 cells were significantly increased by each of toll-like receptor agonists. You can see that in red line for MDA cells and orange line for MCF7 cells. However, in presence of mesenchymal stem cells, you can track that these interactions, blue and green lines. There was a significant reduction in the cytotoxic effects of NK90 cells on MDA cells stimulated with toll-like receptor 3 and on MCF cells stimulated with toll-like receptor 3 and 9 agonists. The cytotoxic effects of toll-like receptor 7 and 9 agonist on MDA cells and toll-like receptor 7 agonist on MCF7 cells were not affected by the presence of mesenchymal stem cells. In conclusion, according to our findings, toll-like receptor 3 and 9 agonists significantly increased the antitumor effects of NK92 cells on breast tumor cells in vitro. However, suppression rather than activation occurred in 92 cells due to the simultaneous induction of the immunosuppressive effects of mesenchymal stem cells by these agonists. On the other hand, the toll-like receptor 7 and 8 agonists provided a low NK92 cell induction. However, the inhibitory effects of MSCs were not triggered. Therefore, toll-like receptor 7 and 8 agonists provide a more significant antitumor activation than toll-like receptor 3 and 9 agonists. In conclusion, our findings suggested that the use of toll-like receptor 7 and 8 agonists to induce antitumor effects of NK cells in a tumor tissue rich in mesenchymal stem cells may be a better approach. This is my last slide. Thank you for attention and patience. I would like to thank the other researchers, especially Professor Dr. Cengiz Kırmaz and Dr. Rabia Bilge Özdemir, who participated in the study, and the TÜBİTAK for supporting our study. Thank you very much.